And welcome to the second segment of our 2017 National Symposium. Thank you so much for joining us. Today you're going to hear from the experts. Today we're going to hear from Dr. Suchowersky, from Dr. Leavitt, and from Dr. Guttman. We are sorry that you won't be able to see Dr. Suchowersky today. We've had a few technical challenges, but she's going to be here with her voice and showing you slides, but we do apologize that you won't be able to see her. We'll start in that order, starting first with Dr. Suchowersky. You're on, Oksana.
Thank you, and welcome everyone. As we're all aware, Huntington's disease is a slowly progressive condition. And while we are able to treat symptoms quite well in many cases, we unfortunately can't really slow down the progression of the condition as yet, although later on you'll be hearing about studies that are attempting to do this. We also cannot easily measure how the Huntington's is progressing without using techniques such as 3 Tesla MRIs or other measures. So the study that I'm going to be telling you about first is just simply called the GM1 study. And GM1, what is this? Well, this is stands for a type of ganglioside, and ganglioside refers to a fatty type of substance that is present in all the cells of our body, particularly in the brain. And it's a combination of a molecule of sugar, and another chemical called ceramide, which combine and are incorporated into all cells, into the membranes of the cells, help keep the cells healthy, help keep them from being damaged, and it also helps in sending messages from one nerve cell to another. So they have multiple functions in keeping nerve cells healthy and sending impulses back and forth. There are a number of gangliosides, and they're just numbered GM1, GM2, GM3, just based on their composition. Now, we also know about the importance of gangliosides because if in someone who has a hereditary deficiency of a ganglioside or a hereditary deficiency in an enzyme which makes the ganglioside, this leads to serious neurological problems. So we know how important they are for normal function. So over the past 10, 15 years, there's been interest in these gangliosides, particularly in GM1, and whether if we give someone extra ganglioside or GM1, we can protect the nerve cells in neurological disorders, and this includes both Huntington's disease and Parkinson's disease, whether this GM1 can help reinforce cells and keep them healthy, and whether in fact GM1 is lower than it should be in people with HD. So this led Dr. Suchowersky to do a study, and Dr. Suchowersky is a basic researcher here at the University of Alberta in Edmonton, to look at mice who have who have been given Huntington's genetically, artificially. So, and what she found is that GM1 is in fact lower than it should be in mice with artificial Huntington's, and that when she replaced or gave them GM1 by an injection into the brain, that in fact the symptoms of Huntington's improved in these mice. And I've seen these mice in her lab, and they are quite, it is sort of quite definite that we see an improvement in not only the motor function, but in also the cognitive function in these mice. So this was a very exciting finding, and it led her to recruit other people at the University of Alberta, including myself, in doing a larger study, which is called the GM1 study. So so the study has two parts to it. The first is a basic research part, which Dr. Scipione is in charge of. And the purpose of this study is to develop a way where we can accurately measure GM1 in the blood and in the spinal, that we can just measure accurately GM1, and also to help determine what exactly GM1 does in the brain and how we can improve that function. The second part of the study is a clinical study of which I'm in charge of, and it is to see whether GM1 is lower than it should be in people who either have symptoms of HD or at risk for developing HD, as compared to controls, to people who don't have HD. So we proposed this study, and we're very fortunate to get funding from Brain Canada and Huntington Society Canada to do this study, mainly at the University of Alberta. So this is called a proof of principle study. So we're looking at to determine, to confirm the hypothesis, whether we can accurately measure GM1 in people, what the levels are normally in people with HD, and also at people at risk for HD, and to see whether the levels change as the condition progresses. So the purpose of this is to see whether we can use GM1 as a biomarker, and what that means is whether we can use it to measure how the condition is progressing. So if it is an easy way to measure disease progression, then we can use it in some of the studies that you'll be hearing about later to see whether different treatments that are being tried can help stabilize or even improve the symptoms. So how are we doing the study? Well, first of all, we're doing the study at 3 places in Canada, as I already mentioned, the University of Alberta and here in Edmonton, at the University of Calgary with Dr. Justina Sarna being in charge of that program, and in the University of Montreal with Dr. Sylvain Chouinard running the Montreal program. And what we're doing, and the study has already started, it's gotten ethics approval at all 3 centers and we're all enrolling patients actively. So all we want to do is to collect blood from individuals, first of all, who have early HD, which means that they've developed symptoms within the last 5 years, with moderate HD, which means that they've developed symptoms between 5 to 10 years ago, and then pre-symptomatic patients who have been tested, who know that they have the gene for HD, but don't have any symptoms. And we're looking for people that we think are going to develop symptoms in the next 5 to 8 years, and also people that have the gene but we think are not going to develop the symptoms for 8, 10 or more years. So we can get a good idea of whether the GM1 decreases over time, when this decrease starts, and that we can accurately measure it. So the blood is going to be drawn within the next year in all participants, and then 2 years from now, so that we can measure the change. We're looking for 20 individuals for each of those 4 groups as volunteers for the study, and then also 80 controls or people who aren't at risk for HD to donate a sample of their blood so we can measure it as well. Now, we're also going to be collecting information which many of you would have normally done at clinical visits, such as the MOCA, the cognitive testing, the UHDRS, the United Huntington's Disease Rating Scale. So we're going to be collecting some basic clinical information along with it. But this study is not meant to take much more time than what you would have during a regular clinic visit, so that it should be easy to participate in. The third part of the study is that once that's done, we want to see whether GM1, we can measure GM1 in spinal fluid from patients with HD, and so we are going to be doing lumbar punctures in about 20 patients, and this is just going to be done at the University of Alberta to collect a little bit of spinal fluid to see whether GM1 is decreased as well. Now, the LP study will be probably combined with HD Clarity, which you're going to be hearing about. I think Dr. Guttman's going to be speaking about it later, that we've been talking to Dr. Ed Wild, who's running this HD Clarity study, about combining resources to see whether we can measure GM1 as well as the things that he is interested in measuring. So we're not going to be doing 2 lumbar punctures in volunteers. We're only going to be doing 1 LP to collect that information. So the GM1 study has several goals, as I mentioned. I mean, obviously, we want to see that we can measure it accurately, that we can see a decrease as the condition progresses, that it's an easy way to measure this progression. And ultimately, we, of course, are interested in doing a clinical study to see whether if GM1 is decreased, that we can add GM1 or give people with HD GM1 either through the spinal fluid or into the ventricle, which is in the center of the brain, and if replacing GM1 will change the course of the progression of Huntington's, whether it will slow down or even reverse any of the symptoms. So this is our ultimate goal. The study I just told you about will take about 3 years to complete, but at the same time, because previous work has shown that GM1 in itself, replacing GM1 in itself, does not seem to have any bad side effects in people, because it has been done in Europe for other conditions, that maybe it can be used to treat the symptoms of HD as well. So that's the GM1 study. Like I said, it is ongoing. We are happy to accept any volunteers, and even if you don't belong to one of the centers that is a part of the study, we may be able to have to develop a method where you can donate a little sample of your blood if you would like. The next study I'd just like to mention, and I think all 3 centers that are online are participating in Legato. Is that correct? That all of us are doing Legato. So just wanted to tell you a little bit about another study that is ongoing across Canada, which we also are very interested about. And Legato is a study that is funded by a company called Teva, which is an international company, pharmaceutical company, which has a medication called Laquinimod, which is not on the market, but it is a medication that is being studied in multiple sclerosis as well as other conditions that are involved with an excess of inflammation in the body. And we now are thinking that in Huntington's disease, that as the cells are affected by the mutant Huntingtin protein, that this may set up inflammation and cause more cell damage. So the thinking is that if we can decrease this inflammation, that we may be able to slow down progression of the condition as well. So Laquinimod, so the Legato study is already underway, and recruitment for the study is completed, and there are a number of patients enrolled across the country with mild symptoms, and the study is with 2 doses of Laquinimod, either 0.5 milligrams or 1 milligram taken 3 times a day, as well as a placebo arm. And I think we had a very good session last year on the importance of placebo in studies, and we always need a group, when we're trying a new medication, we need to know only that we are doing some good. We also need to know that we're not causing any harm. So in this type of study, it's always important to have individuals who are not taking the active compound to make sure that what we think the benefit is actually real and that we're not causing any harm. So this study is a 1-year study. It is going well from what we know so far. All patients, in addition to having a variety of clinical tests as part of the study, also are getting a 3 Tesla MRI. So this is a more powerful type of MRI scan that is present in most hospitals. It's really used as a research tool. And so all patients will be studied both clinically and with this MRI over the 1 year to see whether Laquinimod does help to sort of stabilize the condition or slow down progression. So far, we have not seen a lot of side effects with Laquinimod, although some headaches, pain, and elevated liver enzymes can be seen. But again, we are very interested in this new approach to treatment of Huntington's with decrease of inflammation that may help improve function. So thank you. I will stop there.
Thanks very much, Oksana. That's very exciting, and it's really exciting that the Huntington Society of Canada and the Brain Canada multiple disciplinary program and funding model has been able to fund this project. So many of our community have heard about the Brain Canada funding, so it's great to hear it right from you. So thank you very much for that. Okay, we'll move to Dr. Leavitt for him to talk about some of the clinical trials he's working on.
I'd like to first start off by thanking Bev and the HSC for this opportunity to speak and to talk about some of the research that's going on and some of the trials that are active. I'm going to start with a more general approach to something called Huntingtin Lowering. I think this is relevant to the clinical trial that I'm going to talk about and some of the trials that Mark is going to follow up with. And it's really incredible. This is a super exciting time in research for Huntington's disease. We're really moving into an era of true genetic medicine. We're starting to see therapeutic approaches, treatments that are based on our understanding of the genetics of this disorder and are targeting the real genetic cause of the disease. So that's really incredibly exciting for those of us that have been doing this for many, many years and often supported, as Bev has said, by the Huntington Society of Canada. So thanks for this opportunity and thanks for your long-term support. So Huntingtin Lowering, next slide. This is an approach to treatment of Huntington's disease, and in order to understand it, we have to understand the basic dogma of biology and how we talk a lot about genes and cells and proteins, but in order to understand how Huntingtin lowering works, we need to understand these steps and how the genetic information in DNA, it provides the building blocks for cells. And so DNA is transcribed into RNA, and RNA is the messenger that's then translated into proteins, and proteins are the building blocks of cell. And within our DNA, we have specific genes, and the gene for Huntingtin is a gene, and it makes an important protein called Huntingtin. Next slide. And we know that there's a genetic mutation in the HD gene, the Huntingtin gene, and it's an expansion of 3 of the building blocks of DNA, a C, a A, and a G. And those are the symbols for the nucleotides, cytosine, adenine, and guanine, CAG, and they encode for a specific amino acid in the protein. So next slide. So when the mutation occurs in the Huntington's gene, there's an increased number of these repeats, which create a Huntingtin protein with an increased number of polyglutamines. So this abnormal protein then causes injury to brain cells called neurons. So again, we have the gene, we have an abnormal stretch in the gene, it makes an abnormal stretch of amino acids in the protein, and that's what causes brain cells to become injured. So next slide, the hypothesis on how we can treat this disorder using Huntingtin lowering is basically if you can prevent the production of that abnormal protein, that what we call mutant Huntingtin, that should prevent all of the subsequent pathology and either delay or prevent the onset of Huntington's disease. And we now have a number of tools that allow us to interfere with those steps from gene to protein and to specifically block the production or increase the degradation of this abnormal protein Huntingtin. And that's really what we talk about when we talk about this class of therapies. They're called Huntingtin lowering therapies. Next slide. So when we think about Huntingtin lowering approaches, there's a large number of these that are in development. You can target the Huntingtin DNA itself, and there are things called zinc finger proteins, or you may have heard of genome editing, where we're actually changing the DNA itself. This is very exciting, but it's still many years from clinical development. You can attack the RNA itself, and there's a number of different ways to do that with what are called small interfering RNAs, microRNAs, short hairpin RNAs. And you can also attack it using something called an antisense oligonucleotide. Next. And really, I'm going to be focusing on antisense oligonucleotide approaches because that's what's actually in the clinic today. And that's the trial that I'm going to be talking about, and Mark's going to be talking about some upcoming trials using antisense oligonucleotides. Next slide. So what is an antisense oligonucleotide? An antisense oligonucleotide is basically a small piece of artificial DNA that is complementary, so it binds specifically to an individual RNA, and it has enough of these nucleotides that will only bind to a single RNA in the entire RNAs produced in our cells. So it can be very selective. It only targets the RNA from a single gene, and what it does is when it enters a cell, next, it binds to the RNA specifically. So here's an ASO now bound to the RNA. And when it does that, it causes the cell to recognize this as foreign. And a specific enzyme that we have in our body sees that combination of the antisense oligonucleotide and the RNA and chops up the RNA. Next. So this is something called RNase H. It's an enzyme in the cell, chops up the RNA and prevents it from producing the protein. So that's the basic mechanism. We're co-opting a mechanism in the cell that is already in existence, this RNase H mechanism, and we're using it to cleave and chop up the Huntingtin RNA and prevent production of Huntingtin protein. Next slide. So there's been a lot of basic science. This has been a program that's been going on for over 10 years. And it's taken a long time and an awful lot of work to get this from the laboratory to the clinic. And I'm not going to share a lot of that data, but I'm just going to show you some of the highlights that show the things we had to prove, the community had to prove as a whole to bring these antisense oligonucleotides to the clinic. Now, there's some real important advantages about antisense oligonucleotide approaches that have enabled them to come to the clinic faster than any of the other gene silencing approaches. Next slide. And one of the real advantages is they've been around for a long time. The antisense oligonucleotide approach has actually been used as a therapy since 1998, when it was first approved, when it was directly injected into the eye for a type of eye disease called CMV retinitis. Next. There's also been approved now for systemic injections, so injections intravenously for a type of high cholesterol disease where the antisense oligonucleotides lower cholesterol in this condition. So there are already approved drugs based on this therapy that are out there. There's also been some very important recent developments in neurologic disease. Next. With a new approval of a drug for a type of infantile motor neuron disease. So this is a type of disease that affects the motor neurons, the brain cells that control the muscles, and it causes a paralysis, also called Lou Gehrig's disease or ALS. This is a form that occurs in infants called SMA or spinal muscular atrophy. And an approach based on antisense oligonucleotides has just been approved for SMA. And then there's also next been some important preliminary data and some early phase trials done in adult ALS as well. Next slide. So there's a lot of clinical experience. There's about 20 of these, over 20 of these that are in development. And I just want to briefly again mention this this approach for spinal muscular atrophy, because this uses a very similar approach. The drug is injected into the spinal fluid, and I'll talk a little bit more about that in the Huntington's trial. And this trial was actually stopped early. It was so effective. These patients never develop motor skills once the disease starts. They always show a decline. In individuals treated with this ASO, some of them gained motor skills, some of them are now walking and developing motor milestones that have never been seen before. So a dramatic approach using intrathecal administered ASOs from the same company that's developing this for Huntington's disease. So really very promising and very exciting results just very recently for this approach. Next slide, please. So so ASO drugs have been tested for Huntington's disease, and I'm not going to show a lot of mouse work, but there's a number of things that we wanted to show with these that had to be shown. We had to show that A, they lower the amounts of Huntingtin protein, and this is an example of treatment in a mouse. The blue line represents the Huntingtin treatment. They were treated for a very short period, only a couple of weeks, and yet the Huntingtin levels, as shown by the blue line, are significantly lower than animals that were treated with what's the equivalent of placebo, the black line, for up to 4 months afterwards. So you can administer these drugs even for a short period, and you get a prolonged suppression or decrease of Huntingtin levels in the brain. Next slide. This is shown both at protein and at the RNA level. So it attacks the protein, causes it attacks the RNA, causes the RNA levels to drop, and this causes decreased levels of protein. The bands that are very faint are showing very little Huntingtin protein. The bands that are darker showing more. So you see over a number of months that this effect is prolonged. Next slide. It also improves motor function. This is a standard test of motor function in mice. The blue line again shows the mice treated with the ASO. They are closer to the normal mice, which are the open squares, and much better than the mice that were treated with placebo, the black squares. So next slide. So we can show that treatment in animal models, specifically mouse models of Huntington, show decreased levels of Huntingtin in the brain. They show improvement of some of the signs and symptoms of Huntingtin in the brain. What about a larger brain? How are we going to be able to get this into a large brain like the size of humans? Well, the next best thing we can do is monkeys and pigs. This is an example of an injection into monkeys. Again, it's injected into the spinal fluid, and you can measure that the drug gets, the brown staining is the drug, gets very well into the superficial areas, but not quite so well into the deeper layers. Next slide. And this can be measured, and we can show that, again, over a period of time, from 1, 4 to 8 weeks after injection, that there are significant decreases, mostly in the cortex, the gray matter on the outside of the brain. We get very good knockdown in this slide up in the top right, where it says frontal cortex. Even 8 weeks after the infusion, there's significant knockdown of both protein and RNA out to 8 weeks afterwards. So next slide. So can ASOs be used for Huntingtin? Yes, we've shown that they decrease Huntingtin levels. We show that it improves some of the symptoms of Huntington's disease in these models. There's been a whole series of preclinical, what are called toxicology studies, showing that these approaches are safe, that they don't cause toxicity, Those have all been shown to be safe. And then using the exact same approach, injecting the drug into the spinal fluid, you can show that it gets into brain regions that are affected by Huntington's disease. So this was all shown prior to any studies in human. And there's a lot of hope because these studies all look very promising. And so we moved into the first ever human trial of an approach targeting the genetic basis of Huntington's disease. Next slide. So again, antisense, just to summarize, antisense or Huntingtin lowering therapies target the protein that is, we believe, causes the disease. In the case of an antisense oligonucleotide approach, it binds to the RNA. That RNA-DNA hybrid gets broken down by the cell and prevents the protein from being made. And we can show in a number of models that it does this. Now the big question is, will it do it in people? And the only way to know that is to do human clinical trials. Next study. Next slide. Just a couple of things. Sorry, you can go back. I just wanted to mention a couple of the advantages of this approach that are really important for the safety and for the understanding is that these small little pieces of artificial DNA get into cells very well, and they do have a long-term action for months after an infusion, but importantly, they do wear off and it is reversible, and that's a very important feature when we think about safety, because this is the first time we're actually able to target and lower Huntingtin, and we know that the normal protein actually is an important protein. So the fact that this is reversible and dose dependent is a really important feature. We wouldn't want to knock out all Huntingtin forever using an approach. That could be very dangerous. So that's one of the important safety features of this particular approach, and I wanted to make that point. Next slide. So again, I've talked about giving the drug into the spinal fluid. How is that done? Most people have heard of what's called a spinal tap. A needle is placed between the vertebrae below the level of the spinal cord into a sac of fluid, and that fluid contains cerebral spinal fluid, the fluid that coats the brain. It's produced in the brain. The brain floats in this fluid, and it actually circulates up and down the spinal column. And we can take fluid out, or we can put drugs in, and it will actually equilibrate with the fluid throughout the neuraxis. So if you put it in down here, it will get up to the brain, and the drug will actually penetrate the brain and have direct access to the brain. So these intrathecal infusions are the way that we give these ASO drugs and allow them to get taken up by brain cells directly. Next slide. So this is my last slide, and this is to introduce the Huntingtin, the ASO study that's currently active. This is called the Ionis Huntingtin RX study. Ionis is the company that developed this specific antisense oligonucleotide. It's targeting human Huntingtin. It again went through about 10 years of preclinical development, and this study, the very first patient received the study drug through intrathecal administration in Vancouver in September of 2015. So quite a while ago now. We're almost up to 2 years since the very first patient was treated. Multiple doses of this antisense oligonucleotide have now been tested. The study is closed. All of the patients have been recruited. It's a single site in North America, our site in Vancouver, a number of sites in Germany, and a number of sites in the United Kingdom. All of the study participants have fully been recruited. Most of the patients are completed the study. All of the doses have now been administered, and there's no evidence for any untoward toxic effects, no safety concerns, and we're just waiting for the last few patients to be completed. The study is still blinded. We don't know who got active drug and who got placebo. In this particular study, for every 1 patient on placebo, 3 patients got active drug. So participating gave you a 75% of getting active drug. There were multiple doses in the study. As the study went on, we went to higher and higher doses. We're now at several doses that we think are effective. In this study, we also collected spinal fluid, we collected plasma, we did imaging studies such as the 3T MRI that Oksana mentioned, and a whole wealth of clinical data. But to be very clear, this is a first-in-human study. It's a study for safety of this approach. So far, all evidence suggests that it is safe, and that's fantastic. That's wonderful that we've got to this point where all the doses have been administered, all the patients enrolled, without any concerns about safety. Once the last patient is complete, then we can start to analyze some of this very exciting data. We're very interested in one particular endpoint, which is measuring the amount of Huntingtin in the cerebral spinal fluid, because a number of studies that have been done previously have shown that you can measure the amount of Huntingtin in the cerebral spinal fluid, and that reflects the amount in the brain. So we're hopeful that targeting brain Huntingtin with an antisense oligonucleotide will give us a lowering of Huntingtin in the cerebral spinal fluid. We'll be able to see how the different doses do that, and we'll also be able to see whether there's any effect on clinical measures, the standard testing that everyone does when they come into clinical trials, and on various MRI measures. So that's really where we stand right now, and that's sort of where I wanted to get to with this presentation.
Great. Thank you very much, Blair. It's, our community has been waiting to hear more updates about the trial and everything behind the trial. So thank you very much. It's very exciting indeed. Now we move on to Dr. Guttman.
Thank you very much, Bev. It's an honor to be part of this presentation today, and thank you for allowing me to be at the last, because my colleagues have done a wonderful job and it makes my job a lot easier. Blair, can I borrow your slides for another presentation? They were great. There's a lot of overlap here. Good. So some of the research that I'm going to be talking about is actually research that's going on in many sites across Canada. And the first one I'll mention you've heard of before, called the Enroll-HD study. So Enroll has been going on for 5 years now. It's hard to believe we've been doing it that long. We were one of the first sites in Canada to start up, but both Dr. Suchowersky and Dr. Leavitt's group are involved in many, many sites across the country. And this is what we call an observational study. It does not involve getting medication or getting treatment, but we want to know more about Huntington's, and this is one of the premises behind the study. It's being funded by CHDI, which is a foundation that many of you are aware of. They're a nonprofit organization that's in the States, and their goal is to improve the science and to forward research that will lead to treatment for Huntington's disease, hopefully disease-modifying treatment. Next slide, please. So what is Enroll-HD? Well, Enroll involves a number of assets. One is that we want to speed up the process that you've heard about already. Clinical research is a very complex thing. There are many different steps involved, and one of the slowest steps is actually engaging people like you, people in the community, patients with Huntington's or who have inherited the gene, to get involved. So if we can speed up the process and have basically a list of people that could participate in studies and know all their clinical characteristics, that would be wonderful to improve the process, reduce the time to get to these results that Blair and Oksana have talked about in their own programs. Secondly, we want to understand more about Huntington's disease. So by doing well-characterized research, prospectively, looking at people and following them over time, we can understand how Huntington's actually works. We have experts like Dr. Suchowersky and Dr. Leavitt and myself who spend a lot of time looking after patients with Huntington's disease, but the reality is that at best we're looking after hundreds of patients, not thousands and thousands, which is the kind of numbers that you really need to understand to look at trends and to understand the condition more. So that's the second aspect. We'd like to learn more about Huntington's disease, including many of the genetic aspects, including genetic modifiers that we currently don't know about. And finally, we certainly want to improve the care of people that have Huntington's disease. And I've been co-chair of the Care Improvement Committee to look at data-based information to see why some patients do better than others, and try to understand how we can improve the care while we're looking for a cure to make the lives of people with Huntington's and their families better. Next slide, please. So what is an Enroll? Well, it's really a family study. it's very inclusive. To participate, you can have the gene for Huntington's, either having symptoms or not, pre-symptomatic. You can be at risk and have a family member with Huntington's, either a parent or a sibling, and not want to have testing. You can still be included in the study. You can have had the test and don't have the gene, and that's an important control group. And also there's people who are not part of Huntington's families but are spouses or partners that can participate as well. And having a community control or people that don't have the Huntington's gene is very important for us to understand the research that we're doing. Next slide, please. So over 5 years, we've had a tremendous growth. Some people in the studies have been part of other studies that were smaller studies before this, one called Cohort in North America, another called Registry in Europe. And you can see over 5 years, we're up to 14,000 people that we're following on a regular basis. This is amazing how many people are involved, and not everybody has symptoms. I believe it's about 50% have symptoms in this group, but there are people at risk, people who are in the pre-symptomatic phase. We know from projections looking at recruitment that by the end of next year, by the end of 2018, we're likely to have 20,000 people in the study, which is truly amazing to get information about Huntington's disease. And Europe is just coming online. There have been some challenges with getting all the sites involved, but through the Huntington Society of Canada and strategies that have been going forward in Canada, we've tried to have as many sites involved as we can. Next slide, please. So when you get involved with Enroll, what do? We try to collect information in all the sites for all the patients that are common, so that we have a data set that is rich and is uniform. We also collect blood samples for research, including the genetic modifier research and possible research into biomarkers. And we have both a database and a registry, but also a bioregistry that people who want to do research can get this. They won't know who it's coming from as far as their identity, but we can link the databases so that individuals doing research in drug development or many other aspects can use this, and it's really a shared resource, which is very important for research in our community. Next slide, please. So one of the aspects is to try and improve recruitment into studies, for instance, that Blair and I and Oksana are involved with. And here's an example. So in the studies that have been mentioned in part, so for instance, the Legato study, there's another study called Pride, there's another study called Amaryllis. We tried to use the database to recruit people, and we needed 8,200 people to recruit in the sites involved with Enroll, 815 subjects. So that's a 10-to-1 ratio of having people that possibly could get involved to people who actually get involved in studies. And being involved in studies is not for everybody. There's lots of things that we can talk about of why everybody is not a good candidate, or people may not want to do it, but that ratio is fairly large. So you can imagine if we were getting involved in a time now that we're going to be doing more and more research, we need more and more people, we need a huge database to be able to recruit quickly. And we want to be able to do it better than what we have, a 10-to-1 ratio. We'd like to have closer to a 4 or 5-to-1, and we have to figure out why that ratio is so large. And that's one of the things we'll be working on. Next slide, please. So over the next 5 years, Enroll will be evolving. There is going to be changes, and these are going to be rolled out in the next year or so. One of the things that's very exciting is this RNA lowering strategy and other work on disease-modifying aspects. And for that, we need to have more people at the earlier phase of their Huntington's experience, the at-risk, the pre-manifest, and early-stage Huntington's. So people with more moderate to severe Huntington's may not be good candidates to recruit into clinical trials for a number of reasons. and we're going to be focusing more on these early stage patients for recruitment. There still is lots of research to be done for the more advanced stages, but we're going to make it easier for people, and those will have sort of a lighter version of Enroll to go into. And we want to make it easier as well. We want to look into other things like mobile health applications and maybe even telemedicine to be involved to get people to be recruited easier. So there's lots of changes that are coming up, and hopefully we're going to be able to work together to help with that. Next slide, please. So you've heard a little bit about another study that's ongoing, and both Dr. Leavitt and I are up and running with this HD Clarity, and I understand Dr. Suchowersky is getting involved, but is not actually recruiting yet. So this is a study that Ed Wild has been the major mover from London. Everybody knows Ed from a number of the Huntington Society meetings as well as HD Buzz. So the hope is to develop a biomarker that can be used to measure outcomes. And Huntington's, and Blair has already talked about it. Next slide, please. So one of the objectives is to have, in a large-scale way, to collect spinal fluid of people with Huntington's, both symptomatic, pre-symptomatic individuals, as well as controls, so we can understand some of the biochemistry that's going on, specifically to look at the mutant Huntington's protein that is going to be very important, as Blair has mentioned. There's also generating something that we don't currently have in our biobanks, plasma samples, which are very important for research, and to link that with all the clinical information that we have within Enroll. Next slide, please. We're targeting 600 people that can be in this study. And you can see from this little table that there's going to be people with early and late pre-manifest Huntington's, and we can use special ways of trying to identify that, as well as early, moderate, and late patients with clinical symptoms of Huntington's, as well as healthy controls. So there's going to be a number of sites around the world, and they initially talked about targeting 20 individuals per site. But we've been doing this now for the last 6 months, and we've had a number of people. I think we have 14. It's amazing how many people want to participate. People think of a spinal tap as being something that is, a really traumatic event. But I can tell you that there's a second option to have a second sampling visit, and virtually all of our patients have wanted to come back for a second visit, which I find amazing. Next slide, please. So how do you get involved with this? Well, first of all, you have to be part of Enroll. There's an age range. You have to be capable of agreeing to participate and know what you're getting into and not having any other medical problems. Next slide, please, that could be a problem. So we need to do this in a way that makes sense. Some people, it's just not for them. If they're on any medication that could cause bleeding, obviously that's not a problem. But it's amazing how many people can participate and have participated in this program. Dr. Leavitt was one of the first people with Dr. Wild to help get the initial information to see if spinal fluid measurement of the mutant protein was possible, and the pilot study showed it was. So this is really to validate it. People have a repeat sampling to see about the retest reliability, and we have been told that there will actually be a longitudinal component of the study that will be launched later this year. Good. So in the remaining time, I'd like to talk about another study. Next slide, please. Dr. Leavitt and the team at Ionis, with a number of international sites, was groundbreaking in bringing Huntingtin lowering strategies with ASOs into the clinical arena. And they are far ahead of what this next player is, Wave Life Sciences, to get involved. Wave is a new company. They're new in the Huntington's area. They're new for everything. This is their first study in any neurological condition, but they're dedicated to neurological conditions to do a similar thing to what Dr. Leavitt talked about in the previous Ionis study. So without boring you with excessive details about what the differences are, there are differences in this study, the WAVE study, of how the ASO is targeting what's going on a genetic basis. And in the IONA study, the ASO was targeting what's called the wild-type component of the gene, or the RNA. And in the WAVE study, they are looking to target the allele-specific, so the bad part of the gene that's specifically associated with Huntington's, in a way that directly takes that part out. So we don't know yet, but there is some scientific rationale that that may be a better strategy. Again, we don't know that yet until we try it out. With the WAVE strategy, it will be a bit more personalized than the IONA strategy because people have certain genetic signatures that are called SNPs that people will have to get into the study. So it's not good enough to just have the gene for Huntington's. There is a pre-screening component where people will have to fall into one of two groups called SNP1 or SNP2. There's actually two parallel studies that will be performed simultaneously. And if you don't have that SNP1 or SNP2, you just can't participate because this treatment will not work. So on one hand, it's more personalized, more getting into the exact genetic abnormalities that that individual has, but there may be a fallout that not everybody has this as an option. The other part of the WAVE program is that they're manufacturing these ASOs in a very different way that is called being stereo pure. And the biochemistry behind these ASOs has a lot of chemistry behind it, but suffice it to say that each carbon atom, when it makes bonds to other atoms, can turn left or right. That's called the stereochemistry. And there's hundreds of thousands of permutations and combinations of when these molecules are being manufactured. WAVE has a new approach that creates a stereo pure, a single way that what their drug is going to be manufactured, and there's not going to be lots of different small twists and turns in their molecule. And they think that that would be a better thing. We don't know that until we try. They've done this, the studies in animal models, and they're ready to go into the first in-human studies in a similar way to the IONUS study that's already 2 years out to see if it's safe and effective. So this is a phase 1/2 study looking at safety and tolerability. Next slide, please. So we want to look with this study, how this works. Can we see if it's safe? How do people tolerate this? And it is very, very similar to the study design that Blair participated in. Next slide, please. So there's going to be 4 different treatment groups. There's 2 different components of this. One is that people will have a single dose, and then we'll make sure that everybody in that group, there's going to be 12 in each of the 4 different groups as far as different doses, make sure that everybody's okay, and then they'll have multiple doses after. So each individual will have a total of 4 doses, assuming things go well, and there'll be, or at least there's planned to be, 4 separate doses in a very similar way to the IONUS group. The difference is that in this study, there is the intent of offering open label a real drug. There's a placebo part of this as has been in the IONUS part. The open label will start as soon as each group finishes. So there won't be a gap, at least that's the intention. We don't have all the approvals for that yet, but that's the hope that the company would do this. So this is very exciting. Next slide, please. So how do you get involved in the study? Well, there's a very specific group of people. You have to have early Huntington's disease. You have to be diagnosed. There's an age range involved. It's pretty lenient with respect to medications and things like that. But you have to be stable, and you have to live close by to one of the study centers. So we're one of the first centers that will be up and running. By the time of this broadcast, we're hoping that we'll have the first patients in. And there is negotiation with another Canadian site that has not been fully sorted out. But this will be going on in 15 centers around the world in these 2 parallel studies with SNP1 and SNP2, 48 patients in each group, so 96 altogether. So we're really launching into a time which when I started my research career, is really very futuristic. I would have never imagined us being able to go in and start altering the genetic defects. I started my practice before the gene was discovered. I know Oksana has as well. Blair is just a young pup. He started a bit after the gene was discovered. But this is what we've been hoping for decades. And I think the community is very excited. But I want to make sure that people understand we do not know if this is safe. We do not know if this is effective. We're very buoyed by the information that the IONIS study was safe and the successes in the spinal muscular atrophy program. And we're really hoping this works. But we can't put too much hope until we do these first studies. So that's what I want to say today. I'll hand it back to Bev.
Thanks very much, Mark. I think I have about 9 minutes for questions here. So I'd like to ask you all a couple of questions. First, what one piece of advice would you give to someone wanting to participate in any one of these trials? So why don't we start in order? Why don't we start with Oksana, and then we'll go to Blair, and then we'll go to Mark. Oksana.
Sorry, can you repeat the question?
Sure. What one piece of advice would you give to someone who would like to participate in these trials? Or any one of these trials?
Yeah, I think first of all, you have to know that the trial is completely voluntary and that participation in a trial does not affect your medical care. So whether or not you participate in the trial, you will still receive the same medical care. The second thing is that you need to feel completely comfortable with the study, so make sure that you ask all the questions, ask about all the side effects, and make sure that you know what the potential benefits and the potential risks are before you decide whether or not to participate.
Great.
At the same time, participation is usually, people find it quite rewarding because they do get that sort of extra attention and support while participating in a trial.
Thank you. Blair, do you have anything to add?
Yeah, I mean, I think everything Oksana said is dead on. But importantly, we get this a lot. You know, people may want to participate in one specific trial, but really, I think there's a huge benefit to participating in all research. And so my strong recommendation is that, I make this recommendation for everyone who's interested, you should get involved in a site that does trials, try and be followed there, and you should be involved in whatever trials are available. The best way to participate in the exciting stuff that's coming in the future, and there's going to be many more exciting trials similar to what we're talking about just down the road, is to participate in research today. And really, there is almost a trial for everyone. As Mark very clearly outlined with Enroll, if you want to participate in research, we can usually find something for you to participate in. And Enroll is a great example. So, you know, we generally choose our patients for subsequent trials that are people that have been to the clinic, who get involved in research. So really, the best way to get involved in trials is to get involved with whatever research is available near you. And Enroll is a great starting point.
And a quick question added to that. Do you accept out-of-province participants in your trials?
It obviously will depend on the specific trial. The current WAVE and IONIS trials have a requirement for people to live nearby, as I understand it. Enroll can certainly take individuals from outside. We've done trials, the TRACK-HD trial, which was a very important trial, but also an observational study like Enroll. We had individuals from Puerto Rico, from across the United States, from across Canada coming to our site for that. So certainly, depending on the study, distance may not be an issue, but again, it'll be very site and study specific.
Thank you, that's helpful. And Mark?
Yeah, so certainly Oksana and Blair have stated very clearly the positive aspects. When you're considering participating in a study, you have to look at yourself and say, how much time do you have to spend? Can you make the commitment to do the research? And carefully consider what it all entails. These studies that, the WAVE study, the study with Ionis, they involve frequent study visits, invasive aspects that, many people may have second thoughts about, and I can understand that. In the WAVE study, I believe there's 7 different lumbar punctures over a period of 6 months. That's a lot of activity with needles that some people won't like. So we have to look at how risk averse are you. If you don't like risk, if you don't like being the first person in the world getting a new compound, well, maybe you need to be in an observational study that doesn't have medication being offered. If you want to be on the leading edge, there's opportunities. I totally support having involvement in research. I think that's how, as a community, we're moving forward. And I think we all should be proud of Canada because we're really on the leading edge of so many different things. And when it comes to recruitment into clinical trials, many of the Canadian sites outperform many other countries in the world. So we have a good system. Thanks to Huntingtin Society of Canada and initiatives that are being set up. We have good investigators. We have people that want to come to us as investigators because we know we have a good partnership with our patients. But I feel best when people that I know are being recruited into studies. I know them, they know me, so there's no surprises, and we get to know how it works with each other.
Thank you. So very quickly, and before we close, how important is the HD community to your research? Let's start with you, Mark.
It's the fundamental part that we need. None of us could do clinical research without you. And I think that this is one of the fantastic parts about practicing in Canada. People are very generous with their support. People are engaged. They want to help. Maybe it's part of the Canadian personality. But I think that the relationship that physicians have in Canada with our community, with the Huntington's patient population, Huntington's being a family condition, we have many people that have multiple family members involved in studies at the same time. And Enroll actually envisions that or brings that all together because it is really a family study. So in my opinion, without the Huntington's community, without the families, the caregivers, the patients, everybody involved, and working together in our communities as well with other physicians that we're referring to studies, these are all important ingredients. We do much better than most other countries because of the community that we have here.
Oh, that's good to know. Blair, do you have anything to add to that?
Yeah, I completely agree with what Mark said, but I would also say that the role of the Huntington's community is not just participating in clinical trials. The support that the community has provided through the HSC and through other foundations to push some of the basic science forward is also incredibly critical. We really we have some of the best researchers in the world in Canada. They're supported by this community incredibly well. And we wouldn't be pushing the envelope, as Mark said, if we didn't have that basic support. And that's important at the basic side. It's important to provide us with patients in these studies and for the community to participate there. And it's going to be important moving forward because we're going to need the community to help push our governments, not just to fund the research, but to fund these treatments as they become available. So patient advocacy is going to be a critical issue. We desperately want to get to a point where we're pushing the government to fund effective therapies, and I think we're on the right road. And so the community has been involved in the first 2 phases, and I expect this strong and vocal community to be involved in the 3rd phase when we get there, which hopefully will be very soon.
Thank you. And Oksana, is there anything to add?
No, I think that's great.
Okay. Well, thank you very much to everybody watching today. Thank you for joining in to the second segment of our 2017 National Symposium, and a big thank you to Dr. Suchowersky, to Dr. Leavitt, and to Dr. Guttman. Thank you very much for your time today. Thank you very much for your expertise, and thank you very much for your dedication in the work you do. Together we'll make it happen. Thank you. Have a great day.
Thanks, Bev.
Thank you.